This is FX Radio and I'm Andrew Whitfield-Cook. And with me on the line today is Professor Lyn Griffiths, who is the Executive Director of the Institute of Health and Biomedical Innovation, called IBI, at QUT in Brisbane, leading the research at the Genomics Research Centre, or the GRC. She is a molecular geneticist with over 21 years' experience studying human complex gene disorders with specific expertise in human gene mapping and gene expression analysis, including migraine, cardiovascular disease and several types of cancer. Now that's a mouthful. Lyn, welcome.
Thank you very much, Andrew.
Now, Professor Griffiths, tell me about your research into migraines, because it's a huge area of interest. You led the migraine nutraceutical trial.
I'll start off by prefacing that by saying that I'm actually a molecular geneticist, as you mentioned, and so my interest in migraine was triggered because we were interested in trying to find the genes that play a role in migraine. Mm-hmm. So as most people may be aware, migraine is, for a start, a very common disorder. Affects probably about 12% of the Australian population. There's about 3 times more females than males that suffer from it, so probably about 18% of women and 6% of men. And also children, both males and females are affected. So it's a really common disorder, but the thing to note about migraine is that it runs through families. So if there's someone else in your family who's affected by it, you'll find that there's an increased risk for you personally. But what you tend to find is probably about 90% of people who suffer from migraine have another close relative that also suffers from it.
Wow, that's huge.
So my interest was, it's huge, isn't it? Yeah. And my interest was really at, initially, at trying to identify, clearly if there's a genetic component, what are those genes? Because if you can identify the genes, you should be able to diagnose better, but you should also be able to develop more targeted treatments that's based on the genes that are actually playing a role in it. So your next question was the migraine trial. So, I mentioned that I'm a geneticist. We're interested in trying to track down genes. We have, over a number of years, and we've been working on this for over 15 years now, identified a number of genes that have played a role in migraine. Some of those are genes that relate to hormones, some of those are genes that relate to neurotransmitters, and some of them are genes that relate to blood flow differences, so probably 3 classes of genes. Now, one gene in particular has a mutation in it, and this mutation results in an enzyme that's produced at an abnormally low level if you've got the mutation. Yeah. And what we found that when you actually have this, it occurs much more often in people who suffer from migraine, and we also came up then with a way to target a treatment towards this enzyme defect. And that was the basis of our migraine nutraceutical trial. Yeah.
What are the clinical implications for migraineurs in this nutraceutical trial that you found?
Well, what we found was that probably about 20% of people who suffer from migraine with aura have a mutation in this particular gene that results in a low level of an enzyme. And what we did was undertook a trial that identified the cofactors for this enzyme. And the enzyme's an enzyme called MTHFR, methylene tetrahydrofolate reductase, a really long name, isn't it? And people who have this gene mutation produce about 50% of the level of this enzyme. But there are cofactors that are involved in many different enzymes working. Quite often cofactors can be things like trace elements or they can be vitamins. And in this case, we identified that there were 2 specific vitamins that were cofactors for the enzyme. And then there was another vitamin, and all of these are specific B types of vitamins, and there's another one that fits in a feedback pathway. So we thought, well, if we add more of these cofactors, could we actually make the enzyme work better, even though you've actually got this mutation? And the answer of that, when we actually did our first trial, was yes. Even though you had the mutation, if you add more of these cofactors, you can make the enzyme work faster. Even though it's produced half the level, you can make it work better with the cofactors.
So you can basically push it through.
That's right, you can push it through. But more importantly than that, so we found, first step was, can we overcome the problem with the enzyme? The answer is yes. But the really exciting thing was the people who had this mutation and then went on this trial for 6 months, for those we found that there was a significant drop in migraine frequency and severity and pain associated with migraine. So okay, we could make the enzyme work better, but more importantly, it had this fantastic effect on their migraine. So we actually did a phase 1 trial, which is a trial involving about 50 people, and showed this. But then we thought, well, let's just do a double check and get a bigger number of people, which is what's called a phase 2 trial. Yep. And we got exactly the same results. So over 250 people involved in that, 6 months, double-blind, placebo-controlled trial, and we showed exactly the same thing, that despite having the mutation, we could overcome that mutation and we could have a very significant impact on their migraine frequency and severity and pain.
That's not often seen when you increase the sample size of, or the cohort of a trial. It usually gets watered down, but you got the same sort of results.
Well, truthfully, what you're looking for in a trial is a phase 1 is really just to test whether there's any bad outcomes and whether it's safe to actually use this. So phase 2 is really one where you're looking at efficacy. Does it actually have an impact, and does it have a good impact? And so the outcome from that was yes. The last sort of trial that you tend to do is usually a bigger one, and quite often involves different doses, and that's a phase 3 trial. And we've actually just recently completed a phase 3 trial as well. So we've done a trial involving 600 people. We've had a high dose of the treatments, we've had a medium dose, and we've had a placebo dose as well. I wish I could tell you the outcomes of that, but we haven't quite finished it. So we've actually just finished.
Not yet.
We haven't analysed it yet, but we're hoping that this will... it will have a significant impact for migraineurs, because it will the treatment actually acts as a preventive. It means you take it every day, but from what we've seen in the first 2 trials, it had a very significant impact on reducing how many migraines, how severe they were, how long they last, etc.
Where was this sample size in your initial 2 trials from? Was it a local community thing, or was it multi-centre?
Yeah, luckily we've been working on migraine, as I mentioned, for a number of years, and we're probably one of the first groups in the world to work on it. Australia's been a fantastic place to work on migraine, and I think the reason is that A, it's really common, but B, people are really motivated to help with research on migraine, because there are some very good treatments for migraine, but they're probably only effective in about a third, at most, maybe half of migraine sufferers. So there's a lot of migraine sufferers out there who are really keen at finding a better treatment for them personally. And also I think it's important to try and diagnose in individuals, now that we know that there's multiple genes, which particular gene is it in which person. So rather than trying to give everyone the same treatment that clearly doesn't work for everyone, I think what we need to do down the track is to target treatments to individual gene problems. So I think Australia's been very supportive with this. The Brain Foundation of Australia has also set up a headache register, and a lot of people have been involved in our trials from that. But also the media has actually helped. I think it's because so many people are affected. People would like to do something about it. Whenever we talk to the media and put a call out there for volunteers, we generally get quite a number. So they've all been local. All of the people involved in our trials.
So local Queensland?
Well, not just Queensland, actually. So we've had some people involved in some of our research. The research to try and track down genes has involved people from all around Australia. We have over 3,000 migraine DNA samples we currently work with. We have over 100 large families that suffer from migraine and are part of our studies. We're even doing some studies with Norfolk Island, and we've investigated that for migraine as well. So not just Australia and Norfolk Island.
Are you at a stage yet where you can talk about clinical implications for migraineurs around the world or elsewhere?
Well, I think we can talk about a few things. We can talk about our gene studies and others, which have now shown that migraine is not due to a single gene problem. It's due to multiple genes in different individuals and families. So I think that's made it clear that it's important to diagnose what's happening in your family or in yourself. And then it's also made it clear that treatments are going to need to be targeted towards the individual problems that particular family or person has. So clinical implications of genetics research is that we're starting to get a better handle on the sorts of genes that are involved, the sorts of pathways that are affected, and ideas for treatments and how we can target them. The example I gave you of the MTHFR one is exactly that. It's identifying a gene problem and then working out a treatment that targets that gene and then trialling it to see if it's effective. That's only one. And as I've mentioned, probably about 20% of migraine with aura people have this problem, but there are other genes. There's hormonal genes, there's neurotransmitter genes. So plenty more work to do to try and identify other treatments, but it's starting to become really quite clinical. clinically relevant.
I know this is a lecture unto itself, but can you give us a brief explanation about the MTHFR polymorphisms for the listeners?
Okay, so the MTHFR gene is a gene for the product of the gene is an enzyme, and that enzyme is called the methylene tetrahydrofolate reductase gene. I mentioned the word folate in the middle of that big long name, and so I think that gives you an idea that this particular enzyme is involved in a pathway that relates folate metabolism and what we do with vitamin Bs and folate in our body.
Mm-hmm.
Now, there's a mutation that we initially investigated in relation to migraine that occurs in the MTHFR gene. Some people call mutations polymorphisms, and a polymorphism is really one that just means a change in the DNA sequence. You tend to call it a mutation when it has an effect. So some polymorphisms have no effect whatsoever, and some actually have an effect. So the one that we were particularly interested in is a mutation, and it has a functional effect. So it's not just a change in the DNA, it's a change that results in abnormal levels of the enzyme being produced. Mm-hmm. Now, if you have that mutation, the outcome down the track is an increased level, not just a low enzyme, but an increased level of a biochemical called homocysteine. And in fact, the homocysteine levels produced at the end of this pathway are abnormally high, so it's called hyperhomocystinuria. And it's also been shown that when you have high levels of homocysteine, it increases your risk of stroke. And interestingly, people who have migraine with aura, which is the one we found associated with this mutation, also have an increased risk of stroke. So we think that this gene and the biochemical is playing a role through this pathway in increasing your risk of stroke. And it's thought to disrupt blood flow so it doesn't flow normally, affects endothelial function, etc.
So this has got implications because in cardiovascular disease, hasn't it? Because there's been a whole history of folic acid and high homocysteine in cardiovascular disease, but it's sort of like a prevention thing only. Once you've got cardiovascular disease, giving more folate won't help. How does this translate? Yeah.
Okay, you're absolutely right. So the particular mutation that we've identified in the MTHFR gene was first identified as increasing your risk of cardiovascular disease. And so they found that when you did have this mutation, it increases the levels of homocysteine, and that was found much more commonly in people who tended to suffer from heart attack or from stroke, so cardiovascular events. When we looked at that too, because there was comorbidity between migraine and stroke, that means if you suffer from migraine, particularly migraine with aura, it increases your risk of stroke. Mm-hmm. We thought, well, maybe it's important to look at that particular gene in relation to migraine as well. So that's why we started looking at it. Right. We now know that particular mutation occurs much more often in those who suffer from migraine with aura, and it's not just in Australian populations, it's now been repeated in many different populations around the world, and there's been a big meta-analysis that supports this. Right. So it means that the MTHFR gene is considered to be a susceptibility gene variant for migraine with aura, just like it makes you susceptible to actually CVD-related disorders as well. So if you can drop those homocysteine levels down early, then maybe it's important to actually prevent the development of these. And that's what our thinking was in doing the migraine trial, and that's why we think of this as a preventive treatment. It doesn't totally stop people getting migraine, but it has a big impact on how often and how severe and how common it is. And the idea was with a lot of trials that this might also be important for things like stroke and other CVD-related disorders.
Right,
too.
That's what I was going to ask is, has there been any extrapolations to reducing stroke risk?
Yeah, well, there have been some trials, not quite as exciting as the migraine one. The migraine one so far has been really quite significant. I don't know whether the stroke ones have been quite as significant, but I'm not sure that they haven't looked at a population that might be a bit of a mixed population too. So, at this stage, I think it's not clearly defined yet and probably needs a little bit more work. But certainly with migraine at this stage, it looks really quite interesting.
The story of the MTHFR mutation, some people call polymorphism, I get it, it doesn't stop there, does it? There's different alleles of the MTHFR gene. Is that correct?
Correct, yeah. And there's more than one polymorphism and one mutation in the gene. So, in fact, the gene has many different mutations and polymorphisms. There's one in particular, the one that we first initially showed, which is called C677T, and that's the one that we base the treatment on and also we base the susceptibility on. But subsequently, we and others have also found that there's other variants within the gene or polymorphisms in the gene that are also associated with increased risk. And it seems that if you've got both of them, you have an even greater increased risk or susceptibility to migraine. So a number of these variants actually play a role in it, but I still think that functional one is probably one of the major ones.
So what does this mean for treatment? How do we treat? What difference does having a SNP make to the treatment?
Well, what we found in our trial was that if you think about variations in genes, every person has 2 copies of every gene in their body. So one set of DNA comes from one parent and one comes from the other. So if you can have a mutation point in the gene, you could actually have one single mutation, or you could have 2 copies of the mutation, or you could have no copies of the mutation. And so when we thought about the treatment, that's currently why we're doing a phase 3 trial with the dose arm, because we found in the second trial that people with a single copy of the mutation actually responded better than people with a double copy of the mutation. So it could be that you need a different dose, which makes a lot of sense, doesn't it?
Yeah.
A different dose depending upon whether you got 1 or 2 copies of the mutation. So when you say what difference does having a SNP make? Well, it actually depends whether you've got 1 or 2 copies of this particular SNP.
What about other vitamin supplements that are commonly used in the market, like CoQ10 and Feverfew? Were they looked at, or do they have no relevance to the genetic potential?
Okay, so the CoQ10 and the B1, there's a number of other possible supplements. They don't seem to have any relevance to the actual gene that we're investigating. So at the moment, we've used the cofactors that are known in the pathway, and that's why we've actually included those in that supplement. There have been some interesting results looking at other supplements. So as an example, Feverfew really did have some fantastic results from a trial that was published a number of years ago in the Lancet. So there is some evidence that supports that. But again, it hasn't been continued. It's possibly conflicting with some other ones as well. So sometimes these ones you need to get from just a single phase 1 trial to get them to larger trials before they can actually get out into the market if you want to convince everyone.
Yeah.
The other thing is, there are some additional studies looking at things like magnesium and also, other sorts of trace metal type supplements too, some of which are interesting too. But again, you can't necessarily extrapolate that adding all of these things into one is necessarily the way to go without actually doing the trials.
That's right.
So interesting results, but I can't really say that you should throw them all in there until you've actually done a trial.
No, that's fine. Tell me a little bit about the magnesium research, though.
Well, again, there's some studies that support an increase, that support lower levels of magnesium occurring in migraineurs and perhaps adding more in the diet to reduce the number of migraine. And so there's a number of studies, usually quite small trials, but there are some slightly bigger ones that have supported magnesium as being a useful supplement to reduce the levels of migraine. But it's not something that specifically ties to a gene problem.
Diet has a huge role in maybe triggering a migraine for those or for certain sufferers. Tell me about these biologically active amines.
Right. Well, I think the way I'll come from it is that we've investigated many different migraineurs, over 3,000 migraineurs on our database at the moment. And we have very long and involved sort of questionnaires, and we ask them about what's your migraine like, when did you start getting a migraine, but also what triggers your migraine. And there are very specific and well-known food triggers for migraine. So examples include what I would consider all the good bits, red wine, caffeine, ripe cheeses, chocolate, things like that. So there are some very well-known food triggers, and there's a theory that relates to particular sort of biologically active amines and also foods that contain glutamate. And there are also genes that relate to glutamate as well that have been implicated in migraine. So it makes sense that foods that actually affect pathways in that relate to glutamate, foods that affect pathways that relate to amines make a lot of sense. But again, it's not for all migraineurs. And I think the thing that from our research we've shown it's different genes in different people. So you're not going to have the same triggers for everyone, and you hence shouldn't have the same treatments for everyone too.
And there's no way to tell. There's no link in with the various mutations, the gene mutations?
Well, at this stage, we do diagnostic testing for all of Australia and New Zealand and also parts of Asia. So there are currently 5 known causal migraine genes, and by this we mean they have mutations in them And if you have the mutation, you definitely have the migraine.
Right.
And in this case, they're not probably the sort of migraine that you would think of as being a migraine. They're a subtype of migraine with aura. They're very severe migraine. So 3 of these genes relate to something called familial hemiplegic migraine. This is a autosomal dominant, meaning that if you've got it, every one of your children have a 50% chance of having it. It's a very young age of onset, so usually around about 5 or 6 or maybe 7 years of age, children will get this. It results in very severe migraine. It also has extreme weakness through parts of your body, very common paralysis through half of your body, and also can extend to coma, and even occasionally there's been death as well that's associated with this. This is a migraine. This is due to a gene mutation in 3 different genes. There's also another migraine gene, a potassium channel gene, that causes, again, migraine with aura that runs through families, etc. So there are some known causal genes, and we do the diagnostic test for all of Australia through that. You simply send a blood sample through a neurologist. We do the DNA sequencing. The results go back to the neurologist. So for those, and some of those ones involve calcium channel genes, as well as an ATPase and a sodium channel. And for some of these, there are some treatments that work quite well, but it's important to diagnose it first because it tends to look like other things and can be quite nasty.
Actually,
looks like a stroke, etc.
Yeah, I actually saw one of these migraines when I was doing my nursing training, and it was in a part-time job, and she had a migraine. We thought she was having a stroke. It was very frightening. It
can look like a stroke or it could look like a brain tumour. It's very hard to diagnose and it's very frightening for the patient. So to diagnose it helps them. But as I mentioned, there are some good treatments depending on which gene it is in those people. So it's not that genes are not happening and not being used for diagnosis. They are. Right now, it's only the causal ones, the ones where you definitely have the mutation, you have that disease. The susceptibility ones, like the MTHFR one, are just starting to look at treatments directed to those.
Yeah. Your research also extends, we've mentioned earlier, cardiovascular disease, but also things like multiple sclerosis and cancer. Is there a common link with these methylation mutations? And what does it mean for those that might be a migraine sufferer and have a parent with, say, cardiovascular disease or MS?
Right. The reason that we work on a number of different disorders is actually not because it's the same gene that's involved in all of them. It's actually because it's the same techniques that we use.
Right.
So the sequencing techniques, the using the large families, tracking things through from generations, those sorts of techniques can apply to different disorders. So, it's not a particular gene that pulls it together. It's actually the expertise, the equipment, and the people we're training to be involved in the research. However, going back to your question of methylation variations, there are methylation polymorphisms and potentially a role for epigenetics, which usually relates to methylation and changes in genes too, in relation to migraine, but in relation to cardiovascular disease and certainly in relation to cancer as well. We're currently doing some large-scale epigenetic studies, so looking at methylation patterns across the whole genome and trying to see how that relates from one generation to the next in large families, but also how it relates to disease like cancers and cardiovascular and also migraine.
So this is obviously very early stage, nothing that we can pull out yet or point a finger to look further into?
No, I think it is early stages. I mean, methylation certainly relates to the MTHFR gene, and that's triggered our interest in methylation patterns, but it's not the only gene that has differences in methylation. And so the idea is, for the studies we're currently doing, to instead of just honing in on one gene or another gene, let's do it right across the genome and see which ones, because it's most likely not going to be a single gene. There will be multiple genes.
Sorry to butt in, but that might be very interesting for cancers, for instance, like colon cancer or breast cancer, where the methylation process is highly implicated.
Exactly right. So, and there's a number of studies looking at different types of cancer in relation to that, but I think you can also do that in relation to cardiovascular disease as well. Right.
And is there any particular forms of cardiovascular disease, where there's interest being looked at with a higher
I would say
risk?
The majority of epigenetic studies tend to look at cancer diseases at the moment, but I think that epigenetic patterns will also play a role in neurological type diseases like MS and also potentially in migraine. So we just started that. In terms of cardiovascular disease, it's tricky when you talk about cardiovascular disease because it covers a whole gamut of different areas. So when we think about cardiovascular disease, we are coming at it from the point of view of prevention.
Yeah.
So we tend to look at it as CVD risk traits. So try to identify maybe epigenetic patterns that relate to some of the risk traits like high cholesterol levels, high triglyceride levels, high blood pressure, obesity. All of these things can be identified early on, and if we can identify the genes that play a role, perhaps we can look at gene and environmental information to reduce the development of the end stage, heart attacks and kidney failure, etc.
That raises a very interesting question with regards to the ethics of collection of genes and how are they used to screen, if you like, for diseases and therefore potentially treat. Where are we at in Australia with regards to the ethics or the ethical control of genetic testing, epigenetic treatment, that sort of thing?
So at the moment, as I mentioned before, we do molecular diagnostic testing. So for that, it's just like having any other pathology test done. The way we do it is you go along to your doctor. The doctor looks at you and thinks, we might have a case of a familial hemiplegic migraine here, or we might have an episodic ataxia or something else. So what they'll do is if they think that there's a DNA test available, they'll fill out a pathology form, and tests that we do are listed with the pathology labs. The patient goes along, gets a blood sample. The sample comes to us and we do the testing. And then the results, just like any pathology test, go back to the doctor who then discusses it with their patient.
Yeah.
So the ethics there and the consent is actually something that you do with your doctor. And the testing that we do is something that's considered more of a pathology type of service. In research, to think about what are the ethics involved in gene testing in relation to research, it's different for each particular research project you're looking at. You need to get ethical approval from your own particular university, and you get it for a specific project.
Yeah.
So as an example, we might be doing one looking at migraine genes. So we can't then use those samples to go ahead and look at breast cancer genes. You get ethical approval and consent for a particular project from your institution.
Right.
And universities are very specific about that, about what's allowed and what's not. However, I must say there are a number of companies that are now starting to spring up where you can get gene testing done by taking a swab out of your sample out of your mouth and sending it off to a company, and they will send you back some SNP information. So it really is a kind of grey zone about what's happening commercially in this area. Yes. Not quite what I'm involved in.
I was pointing the finger at Google, don't you?
Well, it's not just Google. There's a number of other companies that are cropping up as well. And I think the problem with this is quite often they're not genes that cause disease. So when I mentioned we do testing for familial hemiplegic migraine, these are causative. We give a result back to the doctor. We're giving a definitive yes or no that's based on whether this is causal in the family. Whereas for that, it's actually not. It's just you might have a 2 times increased risk of getting diabetes over the next 50 years because you have this one SNP. And yet we don't know all the genes for diabetes yet. So it's, to me, seeming a little bit early to be giving that sort of information back to people when we don't know everything yet.
Yeah. So where are we now with the hope of personalised medicine for migraine, like we are with, say, tamoxifen, with certain CYP enzymes, CYP2D6?
Okay, well, I certainly don't think that we've identified all the genes for migraine, which means that we haven't identified all of the personalised treatments that are likely to come out in the future. However, we're making inroads. As I've mentioned, we can now diagnose particular types of migraine through DNA samples, through sequence analysis, and providing that information back to doctors. And for some of these really severe subtypes, there are some very good treatments available once they're diagnosed. So that's one step.
Yeah.
The second step is, again, the MTHFR nutraceutical trial we mentioned. This is one gene, occurs commonly in people who suffer from migraine with aura, and for those with that mutation, this treatment looks like it could be a pretty effective preventive for migraine. So that's certainly personalised medicine. We're not saying that this will work for all migrainists. It's only for those that have the mutation there. And probably just as a last point as well, we've also, and others, identified that not just that hormones play a role in migraine, in some cases for development of migraine in terms of triggering it, but also for causing it, but we've also identified that there are hormone-related genes that play a role in susceptibility to migraine, and we believe that's to this particular hormone. It's called menstrual migraine subtype as well.
Right.
So I think looking at stabilising hormonal levels might be another way we need to think about treatment for migraine as well.
Last symposium, the Bioceutical Symposium 2014, you spoke on your research with the Norfolk Island population. Why did you use the Norfolk Island population for migraine research?
Well, as I mentioned before, we've used a lot of different samples for our migraine research. We have 3,000 individual cases, and we have a lot of matching controls. We also have large families. Now, why would I use Norfolk Island? I'd use Norfolk Island because it's a really unique population that for a geneticist is really something that's quite exciting. So if anyone knows the history of Norfolk Island, I don't know if you do, Andrew, but the majority of people who live on Norfolk Island, that's right, well, not quite, but the majority of people who live on Norfolk Island, about 80% of them, can trace their ancestry back about 12 generations to a particular famous historical event, and that was the mutiny on the Bounty.
A mutiny, that's right.
That's it. And so what happened was that the Bounty was on its way through Tahitian, through the Polynesian islands, and it was heading off to the West Indies. But they stopped around Tahiti. There was a mutiny there. Fletcher Christian and a number of his supporters ended up taking the boat, as well as a number of Tahitian women. And they took it from around that area to a very small island called Pitcairn Island, which is middle of nowhere. It's between New Zealand and South America. This was in the 1780s, they then sunk the boat, just in case anybody was going past and noticed, which they won't. They then sunk it and proceeded to have children, more children, set up a colony, have more children, have a few wars, etc. Yeah. And basically they continued until they were rediscovered in Queen Victoria's time in the 1850s. So it then started to run out of food and water. And so when they were rediscovered, word got back to Queen Victoria. She pardoned the ancestors of the mutineers and gave them Norfolk Island. Norfolk Island was uninhabited, very pretty little island off the coast of Australia. Used to be used as a penal colony for very bad criminals, but it was abandoned because it's a very isolated island, has no natural harbour, and it's hard to get supplies there. And so it became the home of this very interesting population that started from 21 founders and over 12 generations gave rise to the current inhabitants. So for us, we got involved with Norfolk Island in the year 2000. We were very interested in being able to access DNA from people on the island, but also information in relation to disease, because it's what's called an isolated genetic population. It's really mostly one big family. We were able to collect samples and fit most of the people on the island into a very large family with about 6,500 people, right back to the original founders. And we could then use this very large family with really good information that we collected from them to try and hunt for genes. And we undertook studies on migraine and cardiovascular disease, and we've had some successes at finding some of these too.
My next question, it's a very short one, was there's also these implications with regards to, say, cancer risk, or particularly breast cancer risk with regards to the Ashkenazi Jews. Has any research looked at genetic polymorphisms in that group as well, even though they're a lot more widespread?
We've looked at, we've done whole genome SNPs and whole genome expression analysis, and we're currently doing whole genome sequencing of the Norfolk Island population in relation to a number of diseases. Initially, we went in there with a focus on cardiovascular disease and also migraine. We've also got information now, we're doing sequencing in relation to eye disorders like glaucoma. And we're looking at trying to get information in relation to cancers as well, because there's potentially some increased risk, but at this stage, the island actually has limited health support on the island. There's limited screening for disorders like that. There's limited help if people have major cardiovascular or kidney disorders. And in fact, when people do get quite sick with any major disorders, they'll tend to go to Australia or New Zealand. And so it's a bit harder for us to get cancer information, but we're certainly getting as much as we can from all of the inhabitants on the island, and our studies are not finished. So yes, cancer is on the list of things for us to be looking at as well.
I omitted to ask before, what dose of folic acid, and if you use B12, B6, what doses did you use?
Well, we actually, as I mentioned, we did a trial that is different. So we had different doses. So they're quite high levels of folic acid, and it's not currently available across the counter, and you can't buy the tablets. So initially it was 2 mg. We're now using a 1 mg dose. And we had, again, 2 doses of B6 and B12. So it was actually a specially made formulation of all 3 of these.
Okay. So you can get a 5 milligram dose as an S3, as a pharmacist-only dose. Is that too high, or?
It is too high.
Right.
Yeah, I certainly wouldn't be taking 5 milligrams of folic. We, as I mentioned, we had 2 in the original trial, and in the next one we've used 1 mg.
Professor Griffiths, thank you for taking us through your really exciting research areas and the clinical importance of methylation issues and mutations in regards to not just migraines but also some other diseases as well. I'm sure we've all gained much that we can incorporate into clinical practice. So thank you very much for joining us today.
Oh, you're welcome. It was pleasant talking to you, Andrew.
This is FX Radio, and I'm Andrew Whitfield-Cook.